you very much, Iris, for this very kind invitation. It's a pleasure and an honor to be here. So I'm going to switch gear and discuss about infectious diseases. I'm a clinician, as it was said, and also a scientist, and we work on the mechanisms of infection. But of course, we have also a keen interest in genomics. So when I was asked to talk about genome sequencing in infectious disease, I was first not so sure what to talk about. We could talk about sequencing of patients' genome, as we have heard. We could talk about sequencing of clinical samples for microbiological diagnosis, and I will tell you a little bit about this, and mostly, of course, talk about sequencing of microbial genomes. The sequencing of patients' genome in the context of infection, you'd like to detect known primary immunodeficiencies, and as you probably know, there are more than 300 single gene-inborn errors of immunity, and also discover new immunodeficiencies, either by GWAS, whole exome sequencing, or whole genome sequencing. And this is certainly an area in which not only research, but in the clinics, genome sequencing is now being used and is going to be used even more in the future, as we have heard from the previous talk. So what about sequencing of clinical samples for microbiological diagnosis? So here, we are not really talking about genome sequencing, but rather a metagenomic approach to diagnose infection. And there have been many approaches, people who have tried to develop pipelines to use next-generation sequencing to diagnose infection, either to identify known pathogens or to uncover novel pathogens from biological samples. So I'm not going to detail at all the technique. We've been involved in some of these developments over the last years. And just to give you an illustration, in the case of cutaneous granuloma in immunodeficient patient, of unknown origin, we were able, just by sequencing the clinical sample, to retrieve the complete sequence of live rubeola virus vaccine, showing that there is long-term persistence of this vaccine strain, and showing an association between this clinical manifestation and the presence of this unexpected virus here in this example. Same holds true for cases of encephalitis of unknown origin. Here an example of the identification by genome sequencing, high-throughput sequencing of an astrovirus, a new astrovirus, and this was useful to tailor therapy in this patient. And last, I'd like to say on this topic that we are actually moving from research to the clinics, and there is a possibility that in the near future we will use next-generation sequencing and metagenomic approach for first-line diagnosis in infectious disease. So we've conducted, actually my colleague Marc here conducted a clinical trial where we compared the classical microbiological approach to detect infection on NGS from a blood sample taken at the time of the symptoms in patients who were suspected of infection. And just with the conclusion, we found actually that not only next-generation sequencing has a high negative predictive value, so it helps to rule out infection, but it is also at least as efficient in identifying clinically relevant viruses and bacteria than conventional microbiological methods. So, I mean, economically speaking, we are not there, but there is certainly room for the use, the translation of next-generation sequencing in first-line diagnosis in infectious disease. So actually now I'm going to focus on the real topic of my presentation, and I was told, because I was not sure what I was about to talk about, I was told by Joris, you should talk about bacterial genome sequencing. So that's what I'm going to do to focus now. We work on a pathogen called Listeria monocytogenes, and we try to identify the mechanism of infection for this pathogen, but we're also in charge of the surveillance, of the microbiological surveillance of Listeria monocytogenes. which is quite tight in France, as I will explain. So Listeria monocytogenes is a foodborne pathogen. It's a gram-positive facultative intracellular bacillus, which is responsible in human for bacteremia, central nervous system infections, and maternal-neonatal infection. So during the infection, Listeria has the ability to cross the intestinal barrier, disseminate systemically, particularly in immunosuppressed patients, and reach the central nervous system and the placental unit and the fetus. So it has the capacity to cross 3 host barriers. So human listeriosis is a somewhat rare disease. In France, we have 400 new cases per year. But this is a very severe infection, and we've conducted a prospective study to really try to know better the clinical features of this rare infection, and we found this was kind of known, but we clearly proved that there is a huge burden for this infection in terms of mortality. Here you see 30% mortality at 3 months for patients with CNS disease, yes, 46% for patients with septicemia. For women with maternal-fetal listeriosis, there is no maternal mortality, but in more than 95% of the cases, there are complicated pregnancies. So clearly, a severe infection, and you may know that the source of infection of this foodborne infection is very common. These are dairy products and meat products, so the population is widely exposed. There are a few cases, and this is probably due to a combination of bacterial and host factors, as we will see. But there have been not only, and there are sporadic cases, most of the cases in France now are sporadic, but also notable outbreaks. So before surveillance was really implemented, you could have hundreds of cases related to single food source, as shown with this langue de porc en gelée, which is very French. And with the surveillance, the number of outbreaks and the number of cases per outbreak has dramatically decreased. But you see clearly that the food products that have been implicated in these outbreaks are very much associated with a French way of eating. We are talking about cheese, we are talking about delicatessen. But this is not the only country where Listeria is a problem. Indeed, recently, and this is still ongoing, there has been an outbreak in South Africa with a huge number of patients. That's actually the larger outbreak so far, more than 1,000 patients, a very high mortality, 28%. All the country is implicated. You see a huge number of incident cases here, and you see that babies and their mothers are strongly constitute a high number of cases. And in this country, as you know, there is a relatively high incidence of HIV and tuberculosis infection, so there are probably co-infection explaining the relatively young age of those patients. So it's just until the food responsible for the outbreak was identified and was recalled. This is a kind of French bologna, a kind of ham, industrial ham. When the recall took place, then the number decreased and the outbreak was controlled. So a huge outbreak here in a country where systematic surveillance was not implemented, implying that probably surveillance is a good thing. So what can be done regarding Listeria surveillance? What are the goals? These are obvious. Limit the number of listeriosis cases, and thereby its health and economic burden. As you can imagine, the economic burden of such huge outbreaks is really very high. The means is to detect and confirm the cases, detect the clusters of cases related to the same food source, and of course, identify the food sources. The prerequisite, which sounds simple but is not so simple to organize, is to receive as exhaustively as possible both the clinical strains and the food strains in a centralized manner, as we do at the Institut Pasteur. So once you have the strain, you want to confirm the nature of the identification of the bacterium, but you want to type them in the most simple and cheapest way. So there is one relatively simple method that has been developed more than 10 years now, which is Multilocus Sequence Typing applied to Listeria monocytogenes. So what you can do here, this is not whole genome sequencing. You sequence only 7 housekeeping genes which are not too much exposed to the immune system. They do not vary too much among isolates, but still sufficiently to provide some kind of phylogenetic information. You PCR amplify those genes, sequence the PCR fragment, and perform a so-called allelic identification. So you don't look at the whole sequence, but for each sequence type, you ascribe an allele so that the typing of the bacterium is summarized into 7 numbers, and that defines what we call clonal complexes. And if you apply this to a large collection of thousands of strains using this multi-locus sequence typing technique, you see that there are a few major clonal complexes, as shown here by these circles. It defines different lineages. Lineage 1 here, which is a major lineage, as I will come back to. Lineage 2, there is lineage 3, and a small lineage 4 that you don't see in this graph. But you see that there are different serotypes of Listeria, which belong to different lineages and different clonal complexes. So clearly, Listeria is not an homogeneous species, but is made of a series of major clonal complexes. So we asked the question, what was the relative prevalence of Listeria monocytogenes clones in the number of strains we receive on a yearly basis at the National Reference Center in France? These are roughly 2,000 strains per year. So we retrospectively looked at the clonal complex prevalence of strains that we received, either from food and clinical origin. And here you have just this list. I don't expect you to read this, but if you break down this category between clinical and food strains, you see that the repartition, the distribution is clearly different. You have strains, you have clones which are much more prevalent in clinical samples than they are in food samples from which they derive. So there is an uneven distribution of clones in food and clinical samples. Another way of saying this, of showing this, is to see these 2 clones here in blue, which are from lineage 1, which are strongly associated with the food origin, but are rarely identified in the clinics, and other clones like CC1, 2, 6, and 4 here, which are strongly associated with the clinical origin, but not so much prevalent in the food. So this uneven distribution on the following observation was important. So indeed, we showed that there is, it's a bit complicated, but actually fairly simple, an inverse correlation between the so-called clinically associated clones and the immunosuppressive comorbidities of the patients. As I told you, the patients who develop bacteremia and CNS disease have usually immunosuppressive comorbidities, and if you look at the presence of these clinically associated clones or food-associated clones according to the number of immunosuppressive comorbidities, you see an inverse correlation between the clinically associated clones and those immunosuppressive comorbidities as shown here, which together with this uneven distribution strongly suggest that there could be hypervirulent strains. So as you know, an infection is defined by the interaction of host and microbial factors. So you could think of immunodeficiency being a susceptibility factors and virulence on the bacterial side being, of course, a factor for invasiveness of the pathogen. So it could be that the lower the immunodeficiency is, the stronger the virulence here, indicating that virulence is actually heterogeneous in Listeria monocytogenes species. And just in one slide to show you that this is actually the case, when we look in an experimental model of listeriosis, this is a humanized mouse model we have developed, you see in terms of colony-forming units from the spleen and liver and the brain of all infected mice, that these clinically, so-called clinically associated clones are much more neuroinvasive than other clones, for example. So what you can do now, based on these phenotypic differences, is to look at the genome content of those strains, either of clinical or food origin. You can define what is called the core genome, which is the set of genes which are common to all the isolates of the species, and the pangenome, which is the sum of the accessory genomes, or so-called volatile genes. So what you look for here are genes of the accessory genome, which would be associated with hypervirulent clones and could account for the hypervirulence of these strains. So here's just an illustration of this. You have a phylogenetic tree of the different clones. You have the gene content of these different clones. And what you look for here for an hypervirulent clone, which is CC4, is a set of genes which is specifically associated with it, which could be a candidate for being a virulent gene. And indeed, if you knock out this gene and assess the virulence of the mutant as compared to the isogenic parental strain, what you see, and I'm not going into the detail, is a defect in brain invasion in the mutant that misses, that lacks the gene which are specific for this hypervirulent clone. And this can be complemented, so genetically it's working well. So what I just told you is that there is an heterogeneity in the virulence of Listeria monocytogenes defining hypervirulent bacteria and hypovirulent bacteria, and we have the problem of the surveillance. How can we take this into account? So what we look for a good surveillance system in order to control outbreak as soon as possible to prevent a huge peak of cases as observed in South Africa, for example, is to obtain a maximum resolution for strain discrimination, increase the specificity for cluster investigation, detect and control outbreaks as soon as possible, and detect also these hypervirulent clones I told you about. So I told you also about the core genome. So the larger the number of strains you sequence, the smaller the core genome here is, but we defined it as roughly being made of 1,748 genes, which is 53% of Listeria monocytogenes genome. So Listeria is a quite diverse species, and the core genome, of course, is not 100%. So what you can do now is to use the same type of typing technique I told you about, this MLST, except that rather than focusing on 7 genes, you can focus on the core genome, okay? So you will sequence this stuff is going too fast. You will sequence with next generation sequencing the genome of each strain. You will assemble them to make contig, but you don't need to have a full and closed genome. You will look for to detect loci which are part of the core genome, and here you will be doing an allelic attribution so that each sequence variant will define an allele. And you have now a number of 1748 loci that will define CgMLST type. Okay? So in other words, rather than having the 3 megabase complete sequence, you can summarize the genome typing in this relatively small number here. So with this technique, we have a high locus detection rate, we have a low error rate, and most importantly, we have a high congruence with whole genome SNP. And as we will see, SNP, whole genome SNP, is of course very precise, but is problematic in terms of having a nomenclature which is universal and exchanging genomic information from centers to centers. So if we have a simplified method as CgMLST, of course, it's greater. So what we did was to embark into a collaboration with colleagues of other countries which are involved in the surveillance of foodborne pathogens, including Listeria monocytogenes. So these are quite large countries like the US, the UK, Canada, and Denmark, for example, and ourselves in France. And what we did was to sequence and look for the efficacy of CgMLST in typing. We have now in our database more than 20,000 genome. This allows us to study Listeria monocytogenes population biology, and this is important to understand the virulence of the pathogen, as I told you. We have a tool for global genome-based surveillance and a possibility to develop a unified nomenclature. So just briefly, if you now look at these sequences and focus more specifically on strains which have been shown as being part of the same outbreak epidemiologically, what you find is that those, and this is shown in red, those sequences of isolates from a single outbreak usually share less than several allelic differences. So this led us to define empirically a CgMLST type as a set of strain that contains less than 7 allelic differences. And we expect them to be part of the same event epidemiologically, the same outbreak. This may evolve with time and a number of strains and outbreaks investigated. This led us to show, and this is a phylogenetic representation based on MLST, that there is actually international transmission of Listeria monocytogenes. And here you can see circles, I hope you can see circles of several colors indicating that these are made of connected strains from different countries, and this is not only within Europe, but these are also transcontinental transmission. So there is not only cross-country transmission, but international transmission that is ongoing for Listeria monocytogenes. And we could discuss how this happens, but we don't have time really now. So having developed this technique in collaboration with these colleagues, and this technique being quite convenient and allowing exchanges between centers and so on, we decided to test in real life and real time what would be the advantage of using whole genome sequencing for surveillance of Listeria monocytogenes in France, so that we compared it to the gold standard method, which is pulse field gel electrophoresis, which is a whole method I won't detail here. So just a few words about the organization in France. So it's a very centralized system for surveillance. We receive all the clinical isolates. This is a mandatory declaration for listeriosis in France. We also receive all the food isolates which are identified in systematic controls, and also in the context of outbreak investigation. We identify the species by multi-toph mass spectrometry. We check for hemolytic activity, which is a key phenotypic marker for Listeria monocytogenes. We check for antibiotic susceptibility. We do a PCR serogrouping, and then we go for CgMLST, and we also look for the resistant genes and virulent genes in those genome sequences. So this was done first in comparison with PFGE, as I told you. And the results were perfectly clear. I'm not going to detail this. We identified much more types with CgMLSTs and PFGE, as one would expect. The number of isolates per type was significantly smaller. The Simpson index was higher, indicating a better discrimination with whole genome sequencing than PFGE. This is kind of obvious. All this is significant. And if you look at the plateau, PFGE is not so much resolutive. We see CgMLST both from food and human isolates were found from the plateau if it exists. So we have a good typing system. And if you want to see how it shows up, here you have for each histogram a PFGE type, and you see that for each PFGE type you have several MLST type, clearly showing CgMLST type, clearly showing that CgMLST allows to discriminate different clusters within identical profiles. So as I told you, CgMLST allows the identification of a higher number of types. The size of the clusters is significantly smaller, and the source identification is greater. So clearly this is a better method. We can also, and I'm not going to detail too much this busy slide, characterize Listeria monocytogenes resistome and virulence. We can look at antimicrobial resistant genes. This is not an issue actually with Listeria. We can look at resistance to antiseptics, at least genotypically, genes which are involved in resistance to stress, and of course, virulence genes as shown here. So we have now, as I told you, a large amount of data. The clinical isolates are shown in red on this phylogenetic representation. The food and environmental isolates are shown in blue, and you clearly see that some clusters are much more clinical than food, and some clusters are really not associated so much with clinical events. So I told you quite a bit about Listeria, and I'm going to finish in the last minute by other pathogens. Of course, what I told you with Listeria applies to other pathogens. Whole genome sequencing can be used for other foodborne pathogens like Campylobacter jejuni, which is in Western countries the most important cause of foodborne infection, at least in terms of numbers. It also applies to Salmonella. It applies to Escherichia coli and many other pathogens. It also applies to Mycobacterium tuberculosis for prediction of drug susceptibility and resistance. It can be used also for the investigation of nosocomial infection. So clearly genome sequencing is used not for so much for first-line diagnosis now, but for epidemiological purpose. and this is quite fascinating, the information it provides. It also provides information for basic research, which I told you about a little bit. So now I'd like to finish by acknowledging the people who did the work I presented. This is a collaborative work implicating the National Reference Center and WHO Collaborating Center on Listeria at the Institut Pasteur, in close collaboration with Sylvain Brice, who is an expert in microbial evolution. I have also a collaboration with Marc on this use of metagenomics for diagnosis, colleagues from Public Health France, Ministry of Agriculture and PubNet platform, and colleagues from abroad and financing support. And with this, I'd like to thank you for your attention, and I'll be happy to answer any questions.